Hello, I'd like to thank the organisers for this online APRES and everybody for tuning in for this talk about Arachis stenosperma, a source of very strong pest and disease resistances. Well, cultivated peanut itself has a very narrow genetic origin in the polyploidy event in prehistory. This very narrow genetics creates limitations for what can be done, especially for pest and disease resistance breeding using cultivated peanut of pure pedigree. We're interested in bringing in wild genes to broaden the genetic base, and the species here, Arachis stenosperma, the collection was made by José Valls in 1986 near Rondonópolis in Mato Grosso. At the time, the collection site lay some way outside of the city. Valls returned in 2004 and he found remnants of the population, although now embedded within the urban sprawl. We can see it, the plants here spilling out from beyond this little fence with the mango tree in the background. But now this site looks like this. The mango tree has been cut down and no Arachis stenosperma plants can be found. It's now locally extinct. Well, thank goodness for seed banks. The seeds of this succession are maintained and distributed by the USDA. That's a tremendously valuable thing. And it's valuable because of these phenomenal disease resistances. We can see from these slides, the top left slide, when leaves of peanut and Arachis stenosperma are inoculated with the rust spores, the peanut gets an awful lot of lesions, the Arachis stenosperma gets none. It's a similar story with the nematodes at the top right, with the late leaf spot, the bottom left image, and with the fall army worm at the bottom right. Looking at more detail, when Arachis stenosperma is inoculated with juveniles of Meloidigani arenaria, most of those juveniles, almost all of those juveniles, don't manage to penetrate. Those that do rapidly get surrounded by a necrotic reaction. In contrast, when root knot nematode is inoculated on peanut, they rapidly penetrate, form a feeding site and produce egg masses. Now, Arachis stenosperma is, of course, it's an A-genome diploid, and we put it into tetraploid form in 3 different ways, crossing it the same accession of Arachis stenosperma with Arachis batizocoi, Arachis magna, Arachis valida. These are all essentially B genome donors, although Arachis batizocoi, of course, is B genome sensu lato. It's actually a K genome, but it recombines with the B genome. That initial cross was made. The sterile hybrids, cuttings from the sterile hybrids were treated with colchicine, allowed to recover, and some plants double chromosomes and produce seeds. These bat stem tetraploids are crossable with peanut. They were crossed with an elite breeding line from Corley's program to produce F1s and to produce 192 F2 plants. These plants, you can see them here in the greenhouse in Tifton. They were inoculated with nematodes and assayed for disease, galling index, number of eggs per gram of root, reproduction factor. At the same time, they were all genotyped using a SNP array. This allowed the identification of QTLs. And this was really a very satisfying result because we identified 2 very strong loci which reduce the replication of nematodes by substantially more than 95%. And it was also really satisfying because the QTLs coincided with QTLs from a diploid mapping population, which we made some years ago. So that's a really unusual situation of mapping the same pest resistance in both a diploid and a tetraploid genetic background, and the results coming out extremely consistent. Now, all the mapping nowadays is done on the using the reference of sequenced genomes, and that's phenomenally that's really advantageous because for a number of reasons. One is that we can compare to other known resistances. So in this case, we can compare the positions to the main loci which confers resistance in current nematode resistant cultivars like Nematam. And you can see that these stenosperma loci are in a different place. One of them is on the same chromosome, but it's on a completely different arm. So it would be possible to combine these sources of resistance if desired. The other reason that sequenced genomes are so advantageous is that you can reference directly back to the DNA sequence, and that helps developing DNA markers. So we develop DNA markers and use them in marker-assisted selection. We crossed the bat sten with the breeding line and then underwent 3 rounds of backcrossing using DNA markers to select those plants that had the nematode resistant loci and also some other loci that we thought could be involved in other disease resistances. That gave us 300 backcrossed 3 F1 plants. These plants, each of them is predominantly elite genetics with a few pieces of wild genome in them. It's a fantastic resource. They look beautiful. And we could confirm that these backcross plants are nematode resistant. A convenient way to do this is with detached leaves. Each detached leaf can actually be induced to root in vermiculite and then directly inoculated with nematodes. So that's a non-destructive assay, which is really useful when we want to save seeds from all of these plants. Here's the graph showing the replication. The top bars is replication from the control peanuts, and the 0 signals are all from individuals, backcrossed individuals that harbour those resistance loci. This is against Maloidigani arenaria, and we're looking forward to test against Maloidigani hapla. Now, with this backcrossing, you get a recovery of the peanut phenotype, here shown in seeds. Those BC3 lines, they all have seeds which are essentially indistinguishable from cultivated peanut. And here's a couple of lines. You can see it's the same size. Actually, some of them even are a little bit bigger than the recurrent genotype used in the backcrossing. Now, with all of those 300 population, we genotype them using the APRES chip. This is a fantastic tool that allows the profiling of the genome that was developed by Peggy's group in Tifton. Here's a snapshot from part of that. The columns represent individual backcross plants, and on the left-hand side are coordinates, chromosome plus base pair coordinates. And the blue blocks are wild segments of chromosomes. So we have a complete profile for each of those 300 plants. We can view it like this. We can also view it like this, which is the total contribution of wild. Each tiny blue dot on this graph is an individual plant. Quite interesting that some of them actually harbour almost 20%, almost 1/5 wild, but a lot of others harbour much less, as little as 2% or less. And so it's these plants that harbour only a little bit of wild that we're interested in, as long, of course, as they harbour the right bit, which we selected for DNA markers. And this population, we also noticed that it segregates very strongly for late leaf spot resistance and rust resistance. It contains both susceptible plants and highly resistant plants. So for late leaf spot, it appears that these plants have inherited Arachis stenosperma's resistance. What happens with if you inoculate Arachis stenosperma with late leaf spot spores, they germinate, but they sort of wander around over the leaf surface and they don't penetrate or cause disease very different to what happens with Arachis hypergea. We could do a whole genome association. Here each blue dot is a plant, and the areas delimited by red lines are chromosomes, A1 to A10 for Arachis stenosperma, B1 for B10 for Arachis batizocoi, and the lower down the blue dots are, the more resistant is the association with that marker. We can see good marker associations on the A genome with A6 and A4, and that's very satisfying because those regions were identified in a previous diploid population, that same previous diploid population, so that's very satisfying. And there's also a very promising region at the top of B2 for batizocoi. These are candidate regions for conferring resistance against late leaf spot resistance, against late leaf spot. So in summary, Arachis stenosperma, this collection made by Vals in 1986, is a source of multiple pest and disease resistances. We've made it into peanut compatible tetraploid, actually 3 different ones. These will all in due course be submitted to the USDA for multiplication and distribution. Our BC3 population, it's predominantly elite genetics with little contributions from the wild. We know that those wild segments have conferred root knot nematode resistance, late leaf spot resistance, and rust resistance. For the nematode, we have confirmed DNA markers. For the late leaf spot and the rust, we have promising resistance, and the genetic analysis is underway. Well, with that, I'd like to thank everyone for their attention. Thank you very much.